All right, so greetings wherever you are. My name is Big Bright, and that's my contact over there. I'm a bioinformatician by training, and I also do some level of data science. So, one of my friends asked me this question about paternity testing. It has been an issue around, and many people are talking about it as to whether when paternity testing is being done, is it that it is sacrosanct, or there can be issues, there can be some kind of invalid results? Yes, there are some issues that can make paternity testing results not accurate. Yeah, so we want to look at those issues which can make paternity test results inaccurate or invalid. And so let me move straight to my screen, and then we look at what, yeah, so one of them, one of the, I just titled this and I says issues which can invalidate paternity testing. And so I'm going to be, okay, so let me see what okay. So, but before you understand, or before I delve deep into the thing, you need to have some knowledge about the background into paternity testing. What actually leads to paternity testing? So that's what I want to. And so paternity testing, the DNA of both, I just decided to snatch this from my Facebook page. This is an article I wrote on my Facebook page. And I want to tell you the background or the relevant previous knowledge that has to do with paternity testing. What is the knowledge that we have that we are using to be able to use both the genetic material of the father and then the child to be able to determine whether they are, or what the father is the real father or not the real father. And so if you pick the DNA or the genetic material of any organism, there are over 1 million information you can find there. You have alleles, you have genetic marker, you have SNPs, you have short tandem repeats, and then a whole lot of information that is embedded in the genetic material. So one of the information that studies over time has made all geneticists or bioinformaticians to know is that there is something called genetic markers or biological markers, which are constant, which are found in the child and can also be traced to the father. And so what normally happens is, that's why I just put this small text there. You can pause and read it. And that's why I said in paternity testing, the DNA of both alleged father and child is extracted either from blood samples or a buccal swab. Then the specific genetic markers of up to about 15. So you pick the genetic markers from the father, about 15 of them. Then you also pick 15 from the child. And then the markers would have definitely alleles. And then the alleles of those markers are compared head on. So you have picked marker 1 of the father, you pick marker 1 of the child, then you compare them. And now this comparison is weighted, like it is measured, and then given a value called paternity index. Now, which I refer to as PI here, then the total paternity indices that you have weighted is calculated over the 15 markers that you have extracted from the father and extracted from the child. And then the total weighted paternity index is called cumulative paternity index, or combined paternity or percentage index, or whichever way you term it is just all right, CPI. And so when it is being done, it is calculated. And when we realize that the relationship or the comparison has about 99.8% or 99.99, that's what is now in vogue, 99.99% the same, then we can say that the father cannot be excluded as the biological father of the son. Other than that, we say that the father can be excluded as the biological father of the son or the daughter. So this is the background as to how paternity index actually comes in. So you can just read this small text I have put there, and then you are good to go. But it's not always just straightforward like the way I have put it. There are some things that you have to do in the field, in the laboratory, and then the calculations. So in the field, there are errors you can make. In the lab, there are errors that can occur. And then also in interpreting the results or analyzing the results, there can also be errors. And those are the errors that can invalidate or can make paternity testing results inaccurate. And people have done that. People have tested and then they've noticed that the results, they were not satisfied with the results. They go back and test, and then they're actually able to get the actual results. So I'm going to outline some of the things that can make paternity tests invalid or the results inaccurate. So before we do that, I'm going to, so these are the few, look at, we have some, the kind of samples we can pick for paternity testing. You can pick blood samples, you can pick the hair, you can pick teeth, the teeth. You can also pick the buccal swab, that's saliva, like the bone, you can pick a bone marrow, you can pick fingernails, then the tissues from internal organs like the brain, and then the muscles or skin. Now, all those places that I have mentioned, they all contain cells. And any part of the body that contains cells where DNA can be extracted, you can use it for your paternity testing or for your DNA paternity testing. Remember, I've said that there's nothing like DNA tests. That phrase is being used commonly because people resonate with it, but it's not the right terminology. It's DNA paternity testing or paternity testing for short. Now, these are the samples that can be used, but mostly scientists prefer the buccal swab, the saliva, because it's easy, it's less invasive, it does not cut any part of your body, and so many people are very, very, very strict when it comes to using their blood, when you want their blood for any scientific work. So people are not easily yielding to giving out blood because it is invasive. So we resort to what? Saliva. Now, one of the problems that can make DNA results invalid is improper collection and handling of samples. For instance, I just mentioned that buccal swab, like the buccal swab, that is the mouth, you are taking the saliva. Now, if the person has eaten or if the person has taken a drink in very few hours, or sorry, in very few minutes, and then you decide to pick the saliva, you are likely to have your results invalid. So in picking the samples for DNA testing or DNA paternity testing, you make sure that the person hasn't eaten in the last 1 hour or has not taken any drink in the last 1 hour. Both the food and the drink doesn't change the DNA, but it masks it, okay? And that's the reason why scientists don't recommend that. So if you are going to take a buccal swab of someone, you are sure that the person has not eaten in the last 1 hour, and then that is okay to go. That's what can affect it. It will actually mask the DNA. And so when it masks the DNA and then you try to extract or you try to do the comparison, you are likely not to have accurate results. So the results will be inconclusive. Or if it is even there, you might be having a different result either than the actual result because the actual result has been masked, like I just explained. 2, cross-contamination, like putting more than one swab in one envelope. So for instance, you have picked, if you are picking the samples from the individuals, you don't pick just one swab. You can swab the mouth, let's say like 7 to about 5 or 7 to 10 to 15 times. Put each of the swab in different envelopes. Do not put all the swabs in one envelope. Then the other person too, you also make sure that each swab is put in one envelope. Because when you put all of them in the same envelope, there can be cross-contamination, and this can affect the results of your paternity test. Now, the other thing is medical procedure. Medical procedures can, in the case of blood transfusion and then bone marrow transplant, if the person has taken, the people you are going to test them, if they have taken blood, if they have been involved in blood transfusion, they have been transfused with blood, the blood comes with its own genetic material, which is also DNA. Now, it takes time for this DNA to be to be absorbed or to no longer exist. But as long as the person has taken the blood and then the DNA from a different person, so the genetic material of where the blood is coming from or the bone marrow is coming from is within the person, but it's not going to be there for all the time. With time, it will go extinct. But for immediate, when it is not up to like a month or 2 or 3, and then you decide to pick the sample, you are likely to have inconclusive results when you are doing your calculation, because the DNA, the actual genetic material will have two sets of DNA, because the one that you have taken, the blood transfusion you have taken, is still lingering within the person's body system, and then his own blood is also there. And then what is it? That's what happens. That's where even what the paternity testing can be done with a pregnant woman, which I will do a different video and explain how that works. Now, laboratory level errors. There are errors that also occur during the lab session. So lab samples can be mislabeled. For instance, if you've ever been to a lab, you notice that there are too many tubes, too many containers, and I can just put all the test tubes and whatsoever that you can think about what in the lab. They all have labels. So, and you're working with too many samples at the same time, you can mislabel them. So the mislabel, you know what that's going to mean. You're going to pick somebody's results and give to another person. Then genetic material can also be contaminated. DNA handling is the most precocious thing that you have to make sure that you don't joke with. A little contamination can jeopardize the entire result. So if the genetic material, the DNA that you have extracted from either the father or the supposed son or the supposed father is contaminated, once it is contaminated, it will affect all the markers. And then when you start doing the head-on comparison, your paternity index may come down or may not be matching. And remember, it has to match up to about 99.9%. So a little contamination can affect it. And that's it. Now, tampering with the samples. This one is not any scientific thing, but once any of the individuals involved, let's say either the father or the mother or the child, happen to have access to the results, they can tamper with it. Can just put something in it to change the outcome. In a case of maybe the father doesn't want to be proven that I am the biological father, or the child doesn't want to be proven that I am the biological son, they can tamper with the results. But that is if they have access to the result, and this is very rare. It doesn't happen. It's very rare, and there's a report that it has happened before. Now, related alleged father can produce a positive result. Yeah, for instance, this happens when it involves a child who is looking for the father, and the mother decides to say that this person is your father, is your real father, and the child wants to confirm, and then decide to pick the genetic material of the father and himself or herself, and decide to go for a DNA test. If the mother decides to point to someone who is closely related to the father, then the result is likely to look similar or to be the same. And this is mostly when there's a close relationship. People who are from the same father, the same mother. When, like, let's say it is the elder brother who impregnated the father, but the mother pointed to the junior brother. So you can likely have that kind of. But these days, the level of modern technology that we have is very rare that you are able to get the difference, even if they are closely related, except in the case of monozygotic twins. Monozygotic twins are twins which come from the same egg, the same sperm. So fertilized and cleaved, and then the two individuals come out from there. So that's also one of the issues. And then we have something called DNA mutations. Mutations may change in the genetic order or the arrangement of the DNA on the genome. All the DNA in our bodies, they have a pattern that they have arranged. When we say mutation, it means changes. So the way it was arranged, it has changed. And so and remember, the human sperm has DNA. So in the course of maybe coitus, sexual coitus, and where there is fertilization is supposed to, so the man would have contributed his part, which is the sperm. So if the sperm had some kind of a mutant or some kind of mutation, then the mutated strand of the DNA within the sperm that fertilize the mother's egg would be different from the man's real DNA, because the DNA that was contained in the sperm had undergone some kind of mutation before fertilizing the egg. And so when that happens, later on when you test the child may not have exact what a match with the father's DNA, even though the father is the real father. And this happens when it happens in older people or older men, 70 years and above, who are still bearing children, because mutation increases with age. As you are aging, past 60 years, a lot of mutation take place in every part of the body, including the sperms, and that's what happens. So when that happens, you might think that the child is not the real child, but the child is actually the real child. The results can produce different results, just like I've explained. But like I said, these are rare cases. That doesn't happen often. And now there are ways to be able to make sure that all these confounding factors are taken care of. And that's what I mean. So it does happen. And now the issue of what is it, chimerism. This issue is very, very, very serious in genetic analysis, and it also affects paternity test results. Now, to those who might not know what chimerism is all about, chimerism it occurs in an instance where a person, an individual, happen to have two sets of DNA. So the person will have two genes, will have anything that human beings or any organism will have one, they have two. How does it happen? It occurs when like four parent cells. So four parent cells here means that two eggs from the from the mother, two maybe, or with a sperm cell, or with two different sperm cells, what is it, fertilized. And then they decided what is it, the two eggs fuse together to form one individual. It does happen. So two eggs, what is it, were fertilized. So the mother should have born twins, but that's fraternal twins. But in this case, immediately after fertilization, the two eggs fuse together to form only one individual. That person will grow up with what is called chimerism. The person will have two sets of DNA. And because of the two sets of DNA, when you extract his genetic material and compare to the father, you won't find any match. And you may think the father is not, but the father is the one. Now, let me say that this also occurs is not too common, but it does happen. Now, there are algorithms to be able to test chimerism of the two individuals before testing the paternity. So if we test and then realize that the father is chimeric or the child is chimeric, then what we will take care of the chimerism part before we go in for the proper paternity testing. And so these are, and when do these things occur? They occur mostly when you are, it occurs mostly in people who engage in vitro fertilization. So in any of the cases, I tell you an instance where it does happen. So there are women who engage in vitro fertilization. For instance, maybe for some reasons, they have to take the sperm from the man to do in vitro fertilization of the woman. So a child of that sort, or the in vitro fertilization, is likely to result in this chimerism that we are talking about. And so later on, when the child wants to know whether this is my true father, there's likely to be this kind of what a problem occurring there. And so you may think it's not the real father, but that's the real father because of the chimerism. So these are the instances where these things do occur. So I think that's basically how DNA paternity test results can be negatively or can have an invalid result or incorrect outcomes. There are others like reabsorption, where the DNA or the genetic material of the father is missing. Yeah. So immediately after fertilization, the parts that the man has contributed maybe would have undergone something that would have eliminated. It's also due to mutation anyway. And in that case, only the mother's portion would duplicate itself. So one part represents the father and one part represents the mother. But anytime you are testing the paternity, you won't be able to find the father's part, which is there, because the father's part has gone extinct during fertilization or right after fertilization during the process of recombination. So these things do happen. They do happen, and that's why in biology nothing is 100%. So by all that I have said, I'm not saying that when a DNA result is out, and then people are not satisfied, is automatically no. When you are not satisfied, you can go for retesting, and then people will check further, and then to be able to make sure that all these that I have spoken about are not the factors of the results. And if they are, there are ways to be able to make sure that those confounding factors are eliminated, and then the actual results is being produced. The other problem I did not mention is the calculations, the geneticist calculation. There are things that go into the calculation. Maybe I'll do a different video and tell you how to be able to put the weights. I just said that you pick some 15 genetic markers from the parent, and then another one from the child, the supposed child. And then you are supposed to do head-on comparison. You compare one marker, the first marker, to the other, because these are positions in the genome which are known, which we call genetic markers. They are known. So we know them already. So I pick the one that you have, I pick the one that the other one have, and then I match them. So depending on how much the match is, we can give it a weighted average. People have come out with algorithms, but people are also contesting those algorithms. So for instance, if the match is this, and I decide to weight it, let's say 99%, then the other one, I decide to weight it 86%. On what basis did I award this one 99 and this one 80 something? I will do a different video and explains all the nitty-gritties of how the weighting is being done, and how you are able to come out with the overall outcome. So to my very good friend who asked me this question, I think I hope I've explained myself well. And if you like most of this information about genetics and bioinformatics, and please subscribe to my YouTube channel. And by doing that, you are assisting me, you are helping me, and I'm grateful. Thank you. I'll see you some other time. Bye-bye.